All right. Hello, all. I hope you all had a good and safe spring break. Today we're going to be talking, or our lab will be on plant diversity and tree thinking. So I'll give you a brief overview about what that is, remind you of any important vocabulary, and explain the setup of the lab. Before we begin, though, I wanted to briefly discuss what the rest of the semester will look like. And so we didn't have lab right after spring break, and so now we are in the land, plant diversity, and evolution. So the packet is on BBLearn, so make sure you print that out, read it, and also follow up with any of the readings that you're doing for class, because that's going to align up really well with this first lab. After that, we are going to be going into looking at plant tissues, and then we're going to be looking at stomata, and then observing some plant responses or understanding how plants respond to different stimuli. And then we'll go into our last lab, which is angiosperm reproduction, and then we will have our lab practicum that final week. A couple things I wanted to also mention is that we are likely going to assign the full lab report on the plant stomata section. And so over the course of the animal sections, we had been writing parts of lab reports. And so this one is there so you can put all of the feedback that we gave you or your TAs gave you into creating a full lab report. Some more details on that. I just kind of wanted to give you a heads up, and there will not be any, we won't be writing any more things until this full lab report. And then the last thing I wanted to talk about is that the blog post is being pushed back until Friday, April 23rd. And so I believe it was originally due Friday, April 9th, which is the day of your exam. I don't want to overwhelm you all, so let's just push it back a few weeks. We will be uploading the instructions on how to create the blog post in BBLearn soon. And so you'll be able to upload it whenever you want, but we're not going to grade it until obviously Friday, April 23rd. So I hope that helps a little bit. And so if you have any questions about the schedule or what's going to be happening, please let your lab TA know. Okay, so I'm going to go into some quick review about some of phylogenetic terms and remind us a little bit about the things we learned in the animal section. And so one of these things that's important to know is what characters are, and then what the character states can be. And so we looked at a bunch of different characters for the animal lab. And so just remember that the character states can be the presence or absence of something. It can be kind of a numerical thing, so like the number of flower petals, or it could be different categorical data like hair and hair color. And so make sure you review that a little bit or have a good understanding of it. And then the other two things is knowing what a consensus tree is and what a polytomy is. And so we saw in the animal tree thinking lab, we saw what a polytomy is. So a polytomy is a relationship that cannot be fully resolved. And so it kind of looks like this trident here on the tree, and that yes, again, is depicting an unresolved relationship. And so then a consensus tree, move myself. A consensus tree is basically summarizing agreements between multiple trees. So in phylogenetics, it is not uncommon that we will make a lot of trees and then and then summarize them at the end. So it's just basically, you can think of it, I guess, as like the sum of a tree. So we're adding all these multiple trees together to get this consensus tree. Okay. All right, so today in lab, so that was basically the review, or the two things that I really wanted to remind you of. The other things that I will say is that we are going to be talking about a lot of things that have only been mentioned once in lectures, so it might be a good idea to go back and review Dave's lectures on plant diversity or land plant diversity. I know that you guys have gotten through most of it in lecture, so go back and take a look at that. It's really going to help, especially pay attention to the like alternations of generations part. But again, this lab is meant to help you help your understanding of all of these characters that we're going to be looking at. Okay, so brief overview about what today or this lab will look like. So we're going to be exploring plant diversity and evolution through some phylogenetic analyses. So this is cool because in the animal section, we only looked at morphological data, and we will look at morphological data in this lab, but we will also be looking at molecular data. So we'll also give you kind of an insight to how that side of phylogenetics works. And so through exploring the plant diversity and evolution, we're going to be looking at plant morphological data in the same way that we had, or the animal section was set up. So there will be stations around the room for each plant group that you'll be looking at. And your job basically is to go to each section or to each station and decide what the characters are going to be. And then we have a molecular data set for you that is preloaded onto our computers. And so you'll follow some instruction packets on how to analyze that data. And so, again, I kind of already talked about this, but you're going to collect the morphological data from the section or from the stations. And then you will input that morphological data into Mesquite. You will make a tree from just the morphological data, and then you will make a tree from just the molecular data. And then we're going to kind of merge those two data sets and make one consensus tree. And then the last thing is that there will be a write-up on this section. It won't be due at the end of this week, but at the beginning of the next lab. All right, so going into the first part, which is collecting the morphological data. So we have our in-groups, which are going to be our land plants. And so we have lycophytes, angiosperms, mosses, ferns, gymnosperms, and liverworts. So make sure you review this information in your textbook, the lectures that Dave has given, as it'll help you during this lab. The characters that we're going to be looking at are listed. We have 15 characters that we'll be looking at in lab. So it's also important for you to understand what these characters are. And so Dave should have gone through all of these characters in his lectures, so make sure you review those. And we'll go through a few of them just to make sure that you understand them, some of the more tricky ones. But one thing that I like to ask the class is, what is the outgroup or the most ancestral? And then just as a brief reminder, an outgroup can be defined as a baseline organism for comparison, which provides a null hypothesis for comparing character states. So the most ancestral would be everything being a 0. And since you all are not here to answer my question, I'll just give it to you. So green algae is going to be our outgroup for this analysis. So it is not a land plant, therefore it's not going to be our in group, or it's not a part of our in group. And it is going to have ancestral states for all the characters that we have observed. So you can go ahead and write zeros for all of green algae. And yeah, and then after this, I just wanted to briefly go over some of the more tricky characters. And we'll have reminders up in the lab of what these are. And of course, your TAs will be able to answer any of the questions that you have. For the dominant stage of the life cycle, so this is our first character, I like to describe it as basically, what are you likely to see in nature when you go outside and look at this organism? For example, mosses. When you go outside and look at a moss, you're likely to see the green portion of the plant, and that is the gametophyte. So for mosses, the dominant stage of the life cycle is going to be the gametophyte. And so kind of try to step through this process in your head. And then we will also check over your character matrices before you move on, so you'll know if you get something wrong. Don't worry, it's not okay, or it's going to be okay. Free living gametophytes and free living sporophytes is also kind of confusing. So I just want you to remember that free living basically means that they're separated and they develop separately from each other, and they're not dependent for nutrition. The embryo in the archegonium or ovary is pretty self-explanatory, and at all of the stations there will be a slide or something to depict whether or not that is the case. For true leaves, which is character 5, true leaves, roots, and vascular tissue are basically all going to be the same. So true leaves have vascular tissue, and plants that have vascular tissue also have roots. Roots also take up nutrients, so that's an important thing to think about. Microphylls and megaphylls will be indicated at the stations, but definitely go back to Dave's lectures to make sure you understand what the difference between a microphyll and a megaphyll is. Vascular tissue, also an important character to know. Vessel elements will be explained at the stations. And then secondary woody growth is considered growth in girth. So just remember that as well. And then the rest of these should be pretty self-explanatory, but they will also be described at the stations in the room. So I hope that helps a little bit with understanding what these characters are. And so after you go through the stations and fill out that character matrix, you'll have your TA check that. Just make sure everything is A-okay and you got everything right. And then we're gonna move on to working with Mesquite. So Mesquite is the phylogenetic software that we're going to be using in lab today. There is an instructions packet on how to, or basically like a step-by-step of press this button, press this button. That is at the computers. And so you'll work with another partner to go through these instruction packets, and you will input the morphological data and make a tree based on the morphological data. And then you will, in a separate kind of window, analyze a molecular data set. So all of that is explained pretty well in the instructions packet itself. So make sure you read and follow through that. If you have any questions during this section, please feel free to ask your TAs. We've all run through this. So yeah, so we'll be able to answer any questions that you have. The login information for the computers is located on the computer itself. And then one thing I like to tell people, because I understand we're not all math people, is that during this working with Mesquite part of the lab, at the very end, it's going to ask you to take screenshots of the phylogenetic trees that you create. And so on a Mac, you do that by doing Shift Command 4. We will have this up on the board as a reminder. We'll be here to tell you, but just to let you know, for non-Mac users, we'll be using Macs today. So once you do Shift Command 4, you kind of just click and drag where you want to screenshot, and then it'll just pop up on the desktop as a file or as a screenshot file. Again, we'll have reminders in the lab itself. And then again, the part 3, which I already talked about, is just a write-up. So make sure you read through this before you start the activity or even before you come to lab, because you want to be able to talk to your TA and figure out the answers before you leave. And that will be due next week. So I think that is all. We'll see you all in lab.